Okay, today we're going to talk about eukaryotes and prokaryotes. And this is from the book, the biology book, The Big Idea Simply Explained. So, smaller cells should reside inside the larger cells. This is page number 38. So, the first cells on the earth toward this complexity approximately 4 billion years ago. So, first the organic molecules joined together to form a long chain macromolecules, that means large molecules. A primary characteristic of life is its ability to reproduce, and these first molecules would have reproduced by replicating through a series of natural occurring chemical reactions. Okay, we are moving on to the next page. So we are seeing a typical eukaryote and prokaryotic cells, right? Okay, so this is prokaryote and this is eukaryote. What do we have? We have a nucleoid, contains DNA and floats in cytoplasm. And this has nucleus, cell membrane. We have cytoplasm, mitochondria. Okay. Prokaryotes are tiny single cell, so unicellular organisms such as bacteria. The cells have no membrane bound organelles instead of a nucleus. So prokaryotes have no membrane bound cells. What are they? What are membrane bound cells? We have mitochondria. We have mitochondria. We have Golgi apparatus, smooth ear. We have rough ear and smooth ear. What is ER? Endoplasmic reticulum, right? We have Golgi apparatus. All are membrane bound. Organelles. Okay. And while eukaryotes include organisms such as animals, plants, fungi, eukaryotic cells have membrane-bound organelles including a nucleus which contains DNA, and they are much larger than prokaryotic cells. See, as we see. Okay, so this is nucleus. We have nucleus, nucleus. We have Golgi apparatus. We have, these are endoplasmic reticulum, and this is Golgi apparatus. This is Golgi apparatus, and that is endoplasmic reticulum. This is mitochondria. Okay, single cell organism that have no membrane bound cell structures or organelles similar to a modern bacterium. See, Earth atmosphere contained little oxygen at this time, so these first organisms were extremely simple and feeding on abundance of organic molecules and producing energy through fermentation. Like when we hear this fermentation, we think of yeast or maybe a probiotic curd, right? So a process, what is a fermentation? A process that did not require oxygen. Okay, let's see about eukaryotic cells. Photosynthetic bacteria, formerly called blue-green algae, over approximately the next billion years, these photosynthesizers increasingly dominated the living world, releasing oxygen as a waste product. So what is it? The blue-green algae releases oxygen as a waste product. So the Earth's atmosphere and its early shallow oceans experienced a spike in oxygen levels, which had profound effect. Oxygen is highly reactive and can destroy delicate biological structures. So, several prokaryotes evolved mechanisms to deal with this problem, and the most successful of them which was respiration. So they developed respiration, the process of producing energy while converting oxygen into water molecules. What is respiration? The process of producing energy while converting oxygen into water molecules. Okay, we will see prokaryotes and oxygen. Early prokaryotic cells diverged into two distinct lineages called eubacteria and archaebacteria. Archaebacteria and eubacteria. So they have two distinct types. One is eubacteria and one is archaebacteria. Around 3 billion years ago, some eubacteria developed. So what is eubacteria? They developed the ability to convert sunlight into chemical energy, and these were the ancestors of today. The okay, let me pronounce this. Okay, cyanobacteria, a group where is it? I think that is in next page. Where is it? Okay, we are now moving on to the origin of eukaryotes. The evolution of respiration around 2.5 billion years ago could have triggered the development of eukaryotic cells. All advanced life forms contain eukaryotic cells with their more complex internal structure. We have a quote from Len Margulis. Life is a bacterial, and those organisms that are not bacteria have evolved from organisms that were. And Len Margulis are also quoted, "I do not consider my ideas controversial. I consider them right." See, we also see this: a typical eukaryotic cell is about 1,000 times bigger by volume than prokaryote. We have endosymbiotic theory. In 1888, a French botanist Andreas Schimper observed that chloroplast in green plants divided and reproduced in a way that closely resembles the reproduction of free-living cyanobacteria. He suggested that green plants had evolved from close relationship or symbiosis between two organisms. So what can we understand? Sorry. So let me decode what he said. He said that chloroplast in green plants divide and reproduce in a way that reproduction of free living cyanobacteria. So it is similar to reproduction of cyanobacteria. So he suggested that green plants had a close relationship between the cyanobacteria and that. We have a Russian biologist, Konstantin Bereskovsky, was one of the first to notice that there were structural similarities between a plant chloroplast and cyanobacteria. So Schimper said that the green algae, the chloroplast, has symbiotic relationship with cyanobacteria between fungi and algae. So in 1905, the Russian Konstantin, he proposed a proposal that the chloroplast were descended from cyanobacteria that had been engulfed by a host cell and established a symbiotic relationship with it. So, plants owed their ability to photosynthesize to cyanobacteria. This theory that complex organisms arose from the joining of less complex organisms is called endosymbiosis. What is endosymbiosis? The complex organisms arose from joining a less complex organism is called Okay, we will now see a diagram for the theory of endosymbiosis. So first, the two independent bacteria, we have this and this, and next one of them engulfs the other. So one bacterium engulfs the other. As you can see, the circle is now inside one bacteria. So one bacterium lives inside the other. So the internal bacteria are replicated from generation to generation. See, we have from one, and it is replicated from one generation to other generation. And this causes both bacteria benefit from the arrangement. So the theory of endosymbiosis suggests that eukaryotic cells evolved from early prokaryote cells, from being engulfed by other cells and developing a symbiotic relationship. Mitochondria formed when the aerobic bacteria was ingested, and chloroplast formed when the photosynthetic bacteria were ingested. So how the chloroplast formed? Tell me. It is formed when photosynthetic bacteria was ingested by other bacteria, right? And we have how mitochondria formed. So it is formed when aerobic bacteria were ingested by other bacteria. And okay. So this is Lynn Margulies. Okay, we have serial endosymbiosis. Margulies' theory for evolution of eukaryotic cell is often referred to as serial endosymbiotic theory or SET. It is S for serial, E for endosymbiotic, and T for theory. It proposes that eukaryotic cells came about through merging several different kinds of prokaryotic cells. According to Margulies, small bacteria is capable of aerobic respiration. The smaller bacteria which is capable of aerobic respiration is engulfs the larger anaerobic prokaryote cell by borrowing their cell walls. In most cases, this would result in the death of invaded cell. But in some cases, the two cells survived and coexisted. So the parasite, what is parasite?